as awesome. As any questions arise for presenters, feel free to type it in the chat, and then I can relay those questions. We will have, a little bit of time after each presentation to address any questions that do come up. Let's see, I think that's about it. Just a reminder for the presenters. So we're of course looking for a summary of what you did. This is my favorite part of the symposium, to hear what the research that you all did. So thank you for contributing, by the way, to the Tiny Earth mission. I hope that you enjoyed the experience. With that in mind, so we want to hear about the research that you did, but we also want to hear if you could reflect on how the experience was. I know that some of us have had to deal with COVID educational disruptions to various extents. So if you can reflect on any of that and how the research has impacted you broadly. As far as the research that you did, if there's anything that you find unique about your project, or any future directions that you think either you or other students, other researchers involved with Tiny Earth should follow up on, now's a good time to share that. So you'll have 5 minutes to present. Martel is behind the scenes. She's going to be doing the sharing of your PDF. If there's a spot that you want zoomed in on, we can do that. So just kind of guide as needed. And I think if we're ready to go, we will start with Katie. Whenever you're ready.
Okay. Give me 1 second. Hello, my name is Callie Fowler. I am currently an undergraduate student at the University of Wisconsin-Whitewater. Today I will be presenting my research as part of a group collaboration titled Isolation and Investigation of Antibiotic-Producing Bacteria in Wisconsin. With effective antibiotics diminishing and antibiotic resistance rising, a once small infection can turn deadly. This research utilizes Tiny Earth's mission to find new antibiotic-producing bacteria. Over the course of 1 semester, 3 soil samples were collected, isolated, and tested against safe escape bacteria in hopes of observing growth-inhibiting compounds. 8 isolates were found to exhibit this characteristic and were further investigated. The methods of this project followed the standard Tiny Earth protocol, consisting of sterile soil collection, dilutions, creation of master plates, safe escape testing, and gram staining. Figure 1 in the lower left-hand corner depicts dilution starting at 10 to the negative and decreasing by 10-fold on R2A non-selected media. Differential isolates were then identified and patch plated to make master plates. Master plates were then tested against a gram-positive and negative safe escape. Figure 2 in Table 2 in the top center of the column depicts these results. 8 isolates were found to inhibit B. subtilis or S. epidermis, a gram-positive bacteria, or E. coli, a gram-negative bacteria. Purity of isolates was then checked using gram stain methods seen in Figure 4 in the top right-hand corner. These results also provide useful interpretations such as morphology and cell wall structure. Due to limited time, biochemical testing was conducted rather than sequencing. Table 3 and Figure 3 depict these results and observations. Overall, 5 tests were conducted for each isolate. A phenol red broth test provided visual result. Phenol red broth tests provide observations regarding glucose and lactose fermentation and gas production. Visual results can be seen in Figure 3A, showing yellow coloration interpreted as positive fermentation with lack of gas bubbles. Triple iron agar tests were also conducted to observe glucose, lactose, or sucrose fermentation, gas production, and sulfur reduction. Shown in Figure 3B, the left tube shows a yellow-yellow but slant coloration, interpreted as positive glucose, lactose, and sucrose fermentation, while the tube on the right shows a yellow-red but slant coloration, interpreted as positive glucose fermentation. No black coloration was noted in either tube, interpreted as negative sulfur reduction. Sulfur indole motility test was also utilized, showing motility, sulfur reduction, indole reduction, and growth. Figure 3C shows an example of these results, interpreted as positive for motility and growth, but negative for sulfur reduction and indole production due to lack of black coloration. Lastly, catalase activity and oxidase tests were conducted to determine catalase-positive isolates and if the cytochrome C oxidase protein was present. Figure 3D depicts a catalase-positive result indicated by the blue coloration, and Figure 3E shows a positive oxidase result due to observed bubbling. These observational methods were used for every test and for every isolate to assist in interpreting identification. Utilizing Berge's manual of determinative bacteriology, the morphology and the gram stain, and biochemical testing results, isolates were identified as followed: 9A and 7 as bacillus, 20 as brevibacterium, 4 as arthrobacter, 9B as myxobacterialis, 10 as hyphomicrobium, 13 as cellulomonas, 14 as streptomyces. This data contributed to Tiny Earth's large-scale database and helped me better understand the antibiotic-producing bacteria found in each soil sample. In the future, further biochemical testing and sequencing will be done to further support these interpretations. I would like to thank the Tiny Earth Project for continuing their mission and developing this process, and also the University of Wisconsin-Whitewater for providing materials. Thank you.
Great, thank you, Callie. I have a question, a general question. What was your favorite part of the research process?
I think my favorite part was, so I had 2 other group members who decided to not present. And so it was nice to see soil samples from different areas. So I collected mine in Oxford, Wisconsin, which is kind of near the Dells. I had another partner collect near the Milwaukee area and another partner collect in the Whitewater area. So it was kind of nice to see the difference between all of those. I think that was the most fun part.
Yeah, cool. We do have a question. This is from Grace. Which isolate would you like to do further research on? So any favorite or more promising isolates?
So I think I would possibly do isolate 14. Isolate 14 was the one where we saw actual spores on our plating. And we actually saw some residue from it as well. So I think that would be the most interesting one to continue testing on.
Very cool. All right. We have to move on to the next thing. So Justin, when you are ready, go ahead and take it away.
Probably unmute myself first. All right, can you hear me? Okay, everyone, thanks for checking out my presentation. My name is Justin. I'm a senior at the UW Green Bay campus. I'm studying human biology. My research is titled "The Secret is in the Soil" because I believe it's just that. As we know, the rate at which antibiotic resistance is occurring is unsustainable for much of life on Earth currently. If we don't find a way to combat these pathogens soon, common infections that were once just annoying will now be death sentences. That's why I felt it so important to participate in this Tiny Earth project, and I really do believe the secret is somewhere lying in the soil. To start off the experiment, I drove around for a while deciding where to collect soil. After about an hour, I decided to get in the middle of the forest, actually at UW Green Bay. I picked an area that seemed to be full of life, as I knew there was likely bacteria-rich soil. After doing some research in the area, I actually found the soil was Kiwani silt loam soil. Now for the experiment, I first took a gram of my sample and performed serial dilutions that helped me look at individual colonies. I calculated the colony forming units of my sample to be 1.3 times 10 to the 8th power per gram of soil. I found what appeared to be 6 unique isolates. Unique colonies were picked and patched into PDA plates and placed on separate plates, testing for antibiotic activity. 2 of the isolates were found to have antibiotic activity against Staphylococcus epidermis and Escherichia E. coli, I'll just say that. After further review, the 2 isolates, which I thought were different, appeared actually to be the same, so I only pursued one of them. Next, the gram data revealed the isolate to be a rod-shaped gram-negative bacterium. The Conky agar plate revealed the bacterium to be non-lactose fermenting. The catalase test was positive, meaning the bacterium had catalase, which is the enzyme used to break down hydrogen peroxide into water. Beta hemolysis was noted on the blood agar plate. However, these plates were sitting for an extended period of time due to COVID restrictions and Thanksgiving, so I would have much rather liked to read those results within 48 hours, but it's had for about 2 weeks. Lastly, the blast analysis of the 16 srRNA gene was revealed. The bacterium belongs to the genus Pseudomonas with a 99.5% match rate. In conclusion, in the sample I found what seemed to contain a gram-negative bacterium belonging to the genus Pseudomonas, which is supported by the biochemical tests and blast analysis. The bacterium produced antibiotic against both gram-negative and gram-positive pathogens. Thanks for taking the time to listen to my presentation. Are there any questions?
Very cool. What made your final decision about wanting to collect in the middle of the forest? Like, what other areas were you considering?
So I drove around for about an hour, and I was just looking for somewhere that, I don't know, catch my eye, something that seemed interesting. Was there certain plants or certain, I don't know, something that would catch? And I was sick of driving. And I said, I'm just going to walk into the middle of the forest, look for something cool. And that's what I did. I found an area that just seemed to be plentiful with life, something that would potentially have something. And of course, I got the one, but that's how I did it.
Yeah. Were any of you, or Justin, were you here yesterday for the symposium? Did you see Dr. Handelsman, Joe Handelsman's talk?
No.
So she talked about a lot of things, and I believe it was recorded, so I would recommend checking it out, everybody, because she's talking about how she's sharing the cutting edge, like where Tiny Earth is going and the research that's going into analyzing all these samples that you're collecting. And it was super cool. The one thing she was saying is that gram-negative bacteria are really what Tiny Earth is focusing on, or rather the ability to reduce the threat of gram-negative pathogens. So there is a special focus on that. So it's always fun to see that kind of activity. So, all right,
cool.
We have to continue moving on here, but thanks for that, Justin. It's cool. Lily.
Okay. Hi, my name is Lily, and I'm a sophomore at UW-Madison. So my research had to do with tea tree oil, which is a natural antimicrobial agent that has been shown to have bactericidal properties. So we decided to use this to possibly isolate soil bacteria that would produce antibiotics. So the idea was that in a sample with a bunch of bacteria, we would expose them to tea tree oil, and then the ones that survived against it, we thought would be more likely to produce antibiotics when exposed to escape pathogens. So our methods, I did not do this research myself. I did the data analysis, but the methods that took place last year pre-COVID were that soil samples were collected and then diluted so that we could decrease the bacterial concentrations. And then we exposed those bacteria to tea tree oil to isolate specific colonies from a mixed culture. And then we expose it to our pathogen-safe escape relative. And then our metric, we counted the number of colonies that produced a zone of inhibition when exposed to an escape pathogen, and then analyzed that data. So I had 3 different concentrations of tea tree oil that we tested, and those were 1 microliter, 5, and 10 microliters. And then we also had a control group with no tea tree oil treatment at all. And results showed that the control group actually had a lot more antibiotic producers than any of the tea tree oil treatments, either combined as one treatment group or separate, which is the second graph. And then I also did some pairwise comparisons so that, and we found from that when control group was compared to all of the treatments individually, there was a significant difference in the number of antibiotic producing colonies, but when the treatments were compared to each other, there wasn't a significant difference. So from this data, we can conclude that the amount of tea tree oil added to the soil sample doesn't affect the number of CFUs that are antibiotic producers, and the control group also outperformed every treatment group. So that was the opposite of our hypothesis. And we did not find evidence to suggest that tea tree oil is effective in selecting for bacteria, but because past research has shown that tea tree oil has antibiotic properties, it could perhaps be used in concordance with soil bacteria that have been found to be antibiotic producers. So we could do a combination, and there's always room for further research. So I would suggest doing a similar experiment with different antimicrobial agents such as turmeric, honey, or ginger. Some of my other team members did research with those. And these samples were collected in February 2019, and we know how cold it gets. So that reduces microbial diversity in soil. So in further experiments, we could use bacteria samples from locations with warmer climates or from Wisconsin, but during the summer. And I think if I had done this research myself, it perhaps would have yielded different results because I would have collected my soil in like August, September. So it could have produced more antibiotic producers. We don't know, but further research could figure that out. And I would like to thank Dr. P for being such a patient research mentor. And I really enjoyed this experience. It taught me a lot. So thank you.
Awesome. Thank you, Lily. Yeah, I know that, so you weren't there when this samples were originally collected, but of course I was. And yeah, you're right. There is something to the time of, the season in which the soil samples are collected for bacterial diversity. And furthermore, I saw these plates when the tea tree oil was applied to the original soil plates, and that really knocked out a lot of bacteria. So you could see, with your own eyes, the impact of the treatment. And that, so there's the question of how much diversity is that reducing in itself, right, from tea tree oil. Lily, what was your favorite aspect of the project?
I mean, I honestly liked being in lab and going through some of the procedures that we would have done if we had been able to do this all the time. And I liked meeting new people and working with the team.
Yeah, for sure. All right. Well, thank you for that.
Thank you.
JC, when you are ready, the floor is yours.
Can you hear me now? Okay. Good afternoon. My name is JC Jicka, and I currently attend the University of Wisconsin-Green Bay. Today, my presentation is called Digging for Antibiotic-Producing Bacteria in Soil. So over the past several weeks, I've conducted research on soil and antibiotics due to the diminishing supply of effective antibiotics we have today. Specifically, I conducted a study on bacteria found in soil from my father's soybean field. And additionally, my sample collection was near manure runoff from cattle. I dug about 4 inches deep to obtain the O-horizon layer or topsoil layer of soil. And I chose to take from this layer because it is supposed to be rich in organic matter. And also I chose this site because I speculated there would be abundant microbial life present. So first I took my soil contents and diluted them with sterile water in order to obtain individual colonies to calculate colony forming units per gram. The 20 individual colonies of bacteria that grew on my 10 to the minus 4 LB dilution plate were then picked and patched onto PDA and 10% TSA plates. And these plates were called my master plates. My 20 individual isolates were then tested against Staph epidermidis and E. coli escape relatives. And we use these escape relatives because S. epidermidis is gram positive and then E. coli is gram negative. And we did this to see if the isolates produced either broad or narrow spectrum antibiotic activity. And so 3 out of my 20 isolates showed inhibition. Specifically, isolates number 1, number 10, and number 14 showed antibiotic activity against E. coli, but not S. epidermidis. So therefore, all 3 produced narrow spectrum antibiotic activity. To characterize my 3 antibiotic producing isolates, the following procedures were conducted: gram staining, colony PCR, and biochemical characterization analysis. Ethyl acetate extraction was also performed to extract metabolites that have antibiotic activity. So the gram staining resulted in 2 gram-negative rod-shaped bacteria and 1 gram-positive rod-shaped bacterium. The colony PCR test resulted in successful amplification of the 16S rRNA gene in all 3 of the isolates. Furthermore, Yale Core Facility sequenced the 16S rRNA gene for my isolates, and then using BLAST analysis, I analyzed my sequences to find out the genus of my 3 antibiotic-producing isolates. BLAST analysis resulted in isolate number 1 and isolate number 14 identifying as Pseudomonas, and isolate number 10 identifying as Bacillus. So in conclusion, further directions for this project would be to determine the identity of antimicrobial compound for each isolate, and then also, of course, to continue searching for naturally occurring antibiotics in soil samples. Also, this research project really inspired me to pursue a career in a science-based laboratory, and it also opened my eyes to the importance of research to address a worldwide health crisis, especially during times like these. Thank you.
All right, thank you, JC. We have a comment about the cool colors, which I agree with. Did you, the gram stain photos, are those from the internet or did you take those photos?
I actually took them and it was like a struggle. And I definitely wanted to redo them because it's very concentrated and it was really hard to, you know, see the actual individual rod shape. So that I would have redone if we had more time, but due to COVID, we were, you know, only allowed to be in there once a week. But yeah, I thought it was really interesting, the red and like pink color formation of some of the colonies. So yeah.
I think it was worth it. They're great photos.
Oh, thank you.
We did have a question. This is from Todd Kelson. Your master plate was PDA and 10% TSA, but what media did you use to for the actual antibiotic production screening?
So for those, I actually plated, we did 10% TSA plates and PDA for those too, to do testing against our S. epidermidis and E. coli. I tried to do, I just kind of guessed like what would be better if it was PDA or 10% TSA, but we did a couple of each to see what would work better as well. So, but 10% TSA and PDA for those two.
Okay. And another question, were your chemical extract screens successful?
No, they were not. So due to COVID, that test, along with some of the biochemical characterization, they didn't really turn out as great as we would have wanted. And the extractions definitely did not end up working. And I think obviously that could be due to, you know, instead of going 2 days after and looking at the results or continuing the testing, some of the times we had 2 weeks in between there or a week. And so I think that obviously really created a contradiction there. So
yeah, you can see why there's a lot to do in the lab, like when we have the luxury of going in whenever we can, yeah, whenever we want.
Yeah, of course.
That's to our benefit. All right. Well, thank you, JC, for sharing.
Thank you.
Jamie, you're up.
Hi, my name is Jamie Nakonowitz, and I'm going to be presenting my hunt for antibiotic-producing bacteria. In this whole scheme of things, in my class, we were able to actually come into lab every single day, which, wow, that was so awesome. We only missed 1 day of lab, so that's like a win. We did like everything, so it was awesome. I ended up collecting soil from my backyard. I thought there's no way that there are microbes in my backyard for some reason, because why would there be? Brought it to my house. My mom looked at me and was like, what are you doing? I told her what I was doing, and she just kind of looked at me and was like, well, I hope you don't die. So on that note, going through my bacterial isolation, I ended up using my 110 TSA and PDA media. 110 TSA because I wanted to get some just extraordinary organisms coming from it. And I also wanted to use PDA because my professor, Dr. Deborah Davis, told me that PDA was something that a lot of students had gotten antibiotic producing organisms. So I said, all right, what the heck, let's just use them. I ended up using my Shannon-Weaver index values, and I ended up getting a very high amount of species diversity from this. And I also used my Palau's evenness for both of them, which was around 0.89. And that just showed I had a very even soil species sample. With this, I ended up doing my escape screenings. If you go to table 2, my escape screenings ended up resulting in PDA8 and 1/10 TSA7. My PDA8 ended up inhibiting all gram-negative escape pathogens, while my 1/10 TSA was inhibiting both gram-negative and gram-positive, making it a broad spectrum. I was able to go through my biochemical test. I could do basically all of them, which was awesome. I also was able to do my antibiotic resistance test and my preliminary identification. If we go over to tables 3 and 4 for the antibiotic resistance, you can see that out of my 8 commercial use antibiotics, my overall isolates for both of them were pretty resistant to the antibiotics that were chosen. You can also see in table 5 my variety of biochemical tests. For my PCR, I was able to use the 27F and 1492R primers. And from this, I was able to get a sequence for my PDA8. My PDA8 ended up giving me Pseudomonas vulva, which is also known to be a biocontrol agent. Now, unfortunately for my 1/10 TSA7, I was not able to get a sequence for it, but I was, based on my biochemical tests, I was able to determine that it was an allicogene, which does have antibacterial activity. There's just not a lot of information about it. So therefore, this is something that might be novel. So really everything was just very standard, nothing super spectacular about it, other than honestly, the most interesting thing for me was the antibiotics and how resistant both of my isolates were. Something that I'd like to look into the future about would be testing out different soil sites, such as like sandy soil versus clay, maybe even near bodies of water. And I thought it was just really interesting that I was able to educate my parents on soil that did not kill me in the making of this study. So I would also just like to thank Dr. Deborah Davis for helping me just like interpret everything and, keeping everything together. And we got through everything, honestly. So that was like a huge win. So thank you. Thank you so much.
Great. Thank you for sharing. We do have Todd asks, how does your mom feel now since you found potential antibiotics?
You know what? That is such a great question, because my mom looked at me and she goes, so is anyone coming in the backyard to dig up? Like, is anyone going to dig up our backyard now? Like, she kept asking me questions, like every day I got home from school, she just looked at me and was like, so is anyone going to come and dig up our yard? Am I going to get any money for it? What's up with that?
One other comment here. PDA8, so yeah, isolate. PDA8 is cool since it didn't have any activity against GR+, but it hit all GR-. Yeah, so there are definitely cool aspects here, Jamie.
Yes.
Despite trying to downplay it. All right. Thanks again for sharing. Alexis, you are up.
Bitty bitty. All right. Hi, I'm Alexis, and I am a senior at UWGB, and I'm majoring in microbiology and human biology, hopefully finishing up pretty soon. And I'd like to talk to you about what I've been working on for the past couple months. So my identification of antibiotic producing firmicutes started with my soil sample, which I chose to dig up some soil around the Fox River, or not the Fox River, the East River, because I was born and raised here. Everyone hears stories about how gross the East River is. So I definitely wanted to look into that a little bit more. So I gathered about 5 to 10 grams and then used one of those grams to do a serial dilution, and that decreased the population of my bacteria on my plates to a more manageable number. So I was able to calculate the CFU as 5.8 times 10 to the 5th. Then I transferred all of that bacteria from my serial dilutions onto a master plate. And I was able to successfully transfer 20 isolates with a sterile toothpick onto a PDA plate and a 10% TSA plate. Sadly, the bacteria took over my TSA plate, so I had to toss it, but my PDA plate was okay. So I used that as a source of my bacteria for the rest of the experiment. After that, I wanted to look at kind of which ones are gonna produce antibiotics. So I replicated my master plate on LB, PDA, and 10% PDA, or 10% TSA. Then before repeating the pick and patch method to transfer the bacteria, I added liquid cultures of E. coli and S. epidermidis. I used those two as tester strains because they are safe relatives of escape pathogens. I observed zones of inhibition produced by isolates 3, 9, and 15. 3 had broad spectrum properties, while 9 and 15 had narrow spectrum properties to S. epidermidis. I wanted to see the purity of my colonies, so I did a gram stain, and all of my gram stains actually turned up the same as gram-positive rod-shaped bacillus. Even though that they were all the same, they had such unique colonies that were formed. Like they had all the same properties, but they still gathered differently, and I thought that was really interesting. After that, I wanted to do a blast analysis, but to do that, I needed to do a polymerase chain reaction in a solution of nuclease-free H2O, reverse primer, forward primer, and green master mix. This amplified the genetic material of each of my isolates. I sent my polymerase chain reaction to Yale to get sequenced, and they sequenced the 16SR RNA gene of all of my isolates. And then I blast analysis to find out the identification of isolate 3 and 15 belong to the Bacillus genus from the phylum Firmicutes. I wasn't able to sequence isolate 9 because it was too short, so I wasn't able to identify it, but I still kept with it throughout the experiment because I was curious. After that, I did an organic extraction, which I definitely thought was the neatest part of this entire experiment. I had no idea that this was going to be as easy as it was. So I made a large concentration of each of my isolates on separate agar plates and allowed them to grow. And then I cut those agars into pieces to give it a higher surface area, so I could use ethyl acetate and deionized water to organize the different molecules in there. I use ethyl acetate because it's low in density and it tends to attract lower in polarity molecules. So that's going to be secondary metabolites, which usually contain antibiotics. On the bottom layer, I used deionized water, which has a higher density and attracts polar molecules like primary metabolites. So I had 2 layers set up of secondary and primary metabolites, but I wanted to extract the secondary metabolites. So once I did that, I put those into a new glass, let the ethyl acetate dry out. So all of my secondary isolates covered the inside of my glass. And then I used methanol to further extract it because it extracts nonpolar and polar metabolites or materials. And then I placed my isolate and a methanol control group in 4 quadrants on 2 separate agar plates. One of those agar plates, I made an LB top agar with E. coli, and the other one I used S. epidermidis. However, no zones of inhibition were produced, but I was able to identify 2 of my 3 isolates that originally showed antibiotic properties, even though I wasn't able to extract those properties. So I would like to thank Dr. Brian Merkel and the Tiny Earth community for letting me be a part of your project. Thank you so much.
Great, thanks, Alexis.
No problem, thank you.
You're not alone. The activity in the folks, Martel and Martel is one of the folks that do all the processing of all these samples in the chemistry hub. And I'm sure that you're not alone with failing to see activity in the extract itself. It's a common issue. Did you accidentally get a whiff of the ethyl acetate?
I didn't. I stayed as far away from that as I could because I'm one of those people that passes out from formaldehyde. So I was a little worried.
We do have a question here. Who from Grace. Did you look to see if isolate 3 could kill isolates 9 and 15?
I didn't, but that would be a really interesting perspective on things. That would be really cool to do.
Yeah, I agree. All right. On to our last one. So thanks again, Alexis, for sharing.
Thank you.
And Austin, bringing it up in the caboose here.
Hi, everyone. I'm Austin. I'm a senior at UWGB. Just want to start off by saying thank you to Tiny Earth for such a great opportunity and allowing us to participate in the symposium. Starting with my sample site, I checked multiple spots, various different dig sites where I did not think the soil was up to par for the project. Finally came across an area near a pond on the UWGB campus Arboretum Trail just after a rainstorm. And I thought these two conditions would be very beneficial to the project because ponds are generally filled with microorganisms, and a healthy ecosystem like this also likely to produce an abundance of bacteria that would likely be antibiotic producers. Serial dilution is an important step of this project because it allows for better examination of the cells when they are plated. Instead of looking at a plate that is highly concentrated with possibly millions of bacteria, this procedure allows us to get an approximation of the number of colony forming units. The suspended soil solution was plated on an LB auger with cyclohexamide, and the results showed that colony forming units of the fourth plate. Sorry, I had COVID, so my throat's kind of a little bit, which should be 1 to the 10,000 serial dilution, approximately 45 million viable cells. Cyclohexamide is used for this portion of the project because it inhibits translation by eukaryotes. This is important for a soil sample that is strictly looking for soil bacteria due to the fact that fungi belong to the eukaryotic classification, and the growth of fungi from the soil sample onto the plates would not be beneficial. Master plates were made from the diluted plates, and I found that bacteria from my soil sample had the best growth on PDA plates. I think the PDA plate worked better than the TSA plate because it created weird and intriguing growth patterns of bacteria in comparison to the 10% TSA plate, which seemed like a more basic growth that did not go past like the X mark from the pick and patch method. Why this growth seemed to happen like this, I'm not so sure, because both media have proper energy sources within the media for bacterial growth. For the tester strains, E. coli and Staph epidermidis were used for antibiotic production. These two strains were chosen because they are safe relatives of 2 of the escape pathogens. These 2 safe relatives were also chosen because Staph epidermidis is gram-positive and E. coli is gram-negative. When testing for antibiotic production, none of the isolates shows were broad-spectrum antibiotic resistant. After testing for antibiotic production, multiple gram stains were done on all 3 isolates to determine if the bacteria were gram-negative or positive, and also if they were rod or cocci shaped. The results I obtained from the gram stains were 2 gram-positive rod-shaped bacilli and 1 gram-negative rod-shaped bacteria. Colony polymerase chain reaction was then done in lab and then sent to Yale core facility for sequencing. The primers amplified the region of the 16SR RNA gene, which is used as a fingerprint for identifying the genus of bacteria. Once the results from the application were complete, we used a blast analysis database to encrypt the amino acid sequence obtained from the PCR, and this allowed for accurate results of the bacteria's genus. 2 gram-positive bacilli both had a 99.72 match with the Bacillus genus of bacteria. The gram-negative species had a 99.43% match with the genus Pseudomonas. And then the last portion of the project was to do an organic extraction and other biochemical characterization tests, which would assure that the results obtained from the amplification of the 16S RNA sequence was correct. Fortunately, I was not able to obtain all the results due to missing a week of lab with COVID absence. I was able to do a catalase test, which all 3 came back catalase positive. Doing some research on the Bacillus and Pseudomonas genus, I was able to get an idea of what the results would have been if I obtained the other characterization test. Aside from all the challenges that were presented during the semester, this was such a great opportunity to be a part of and such a big learning experience that I know I'll look back on in the future.
All right. Well, thanks for sharing, Austin, and I'm glad to hear that you're persevering despite COVID.
Thank you.
What was your favorite part of the project?
I really liked, because I did a couple master plates, and I thought it was interesting how the bacteria, they kind of like, some of them were kind of gave the same results when you plated them 2 times. like the same growth patterns, but some were different. I thought that was interesting.
Yeah, isn't it? I've seen that before for sure. Very cool. Well, that concludes our presentations. I hope you all enjoyed, and I'm sure you saw the commonalities, right, across the work that was done, but also some subtle differences across different institutions and different choices that you made, right, as individual students and researchers. So thanks again for contributing to the project. Do hope you stick around and attend the next session, which is happening in 15 minutes. So there's a little break, and then we're going to hear a keynote presentation. So with that, I will let us go and have this break. And thanks again from Tiny Earth.